Okay, so here we have annotation of diagrams of seminiferous tubules, so that'd be in the males, and ovary to show the stages of gametogenesis. That's this picture. You could get one just like this. They might ask you to label it. Again, annotation of it means label it or tell me about the parts that they've labeled, A, B, C, D. So here you have a follicle, right? Here you have a stages of gametogenesis. Genesis means beginning, beginning of gametes. They don't, again, I wish they would give a specific list, but the stages they're talking about here, I think must mean this. When you talk about an egg cell that gets fertilized by a sperm, I'm gonna see this is not quite like it shows in this diagram in reality. So I don't know how specific they want you to be, but I'm gonna back it up. When you have a diploid cell, and that diploid cell might divide by mitosis to make a lot more of itself. That diploid cell is inside a follicle here, that compartment that's called a follicle, right? If it undergoes meiosis to make gametes, the thing now is going to be called a primary oocyte. This has gotten a message. What kind of message? Some kind of signal molecules that it's time for you to do meiosis and not more mitosis to just make more of yourself. So this diploid cell now is going to start prophase 1 of meiosis. And in that, if you remember, that's when homologous chromosomes already doubled up because S phase has happened already, are going to find each other and pair up in a tetrad. We get a review on all that stuff, right? That process is called synapsis, and that is the key difference between mitosis and meiosis, is this pairing, and then later separation of homologous partners in meiosis 1, right? When meiosis 1 is completed, then we have two cells that each have a haploid set of chromosomes that are still double-stranded, but now they have the infamous one from each pair, right? One of each kind, one from each pair. That's what meiosis 1 accomplishes that mitosis never does, is to cut in half the number of chromosomes, all right? But now you notice this. What happens here is that the cytokinesis of this cell, the actual splitting of the cell, is not equal in terms of cytoplasm. It is in terms of what's in the nucleus. Each of these have one set of chromosomes, like we said. But this one gets almost all the cytoplasm. And this one, which is for some reason called a polar body, this one here is just not going to make it anymore. It's not going to, without cytoplasm, it's not going to live any longer. It might divide into two more polar bodies, but it probably won't even that. This one, however, is going to continue through meiosis 2, and it then is going to also have an unequal cytoplasmic separation or division, and only one cell out of a potential 4, right? Meiosis has one cell divide twice to make 4. Only one of those potential 4 is actually going to be a fertilizable egg cell. All right, I'm going to come talk about this later, but I don't know how specific they would be. In actuality, what gets released from an ovary is this thing, and what gets fertilized is this thing. So showing this little sperm cell fertilized, that is not quite accurate. But these stages and their names, I would think, is what they mean by stages of gametogenesis. These they might ask you to identify in a diagram like this, right? And then the final thing that we would call an egg cell, ovum is a synonym for egg cell. It's not actually what gets fertilized, but I think in most simplified versions, they would talk about this being fertilized. Anyway, back to this diagram, right? Which they might ask you to annotate a diagram of that, right? So here's that happening in all these stages. Inside this growing follicle, this meiosis is happening until finally what this shows being released is a secondary oocyte. And I would go with that, but I am again positive that that is also what gets fertilized. Remember, I said this is what gets released from an ovary, and it also gets fertilized. This meiosis 2 doesn't actually happen unless fertilization happens, which means if a female, this is mostly mammal stuff. If a female or actually I would say vertebrates, not just mammals, but reptiles and birds also. If a female never has a secondary oocyte fertilized, then that means she's never had a cell complete meiosis. Because again, this one still has to go through meiosis 2 before it gets down to a single set of single-stranded chromosomes, which is what the cells after meiosis 2 have. So this ruptured follicle again is now called a different name. They would want you to annotate that or identify it in a diagram. So that's all that. And I wish they'd have been more specific about parts they wanted you to do. So now we go with this. So we just showed this on the last diagram. Maybe I should have put this on the last diagram. That the genesis of sperm cells and egg cells both involve mitosis, and that is to take those diploid germ cells that are in a follicle in an ovary or inside these tubes, which we'll see in a second in a male. It's called seminiferous tubules, to cause them to make more of themselves. Otherwise, they would kind of run out, especially in a male, as you'll see. But then some of them, again, get a signal to not do more mitosis, but do meiosis, become different. And so all those words got in here without too much specifics about what else they might ask you about them. So the processes of sperm generation and egg generation result in different numbers of gametes, like I said before, with different amounts of cytoplasm. We already talked about that, at least with the egg part, and once we get a sperm cell diagram up here in front of us, we'll come back to and talk about that. But also timing, which I'm going to add, and I don't remember that they add in here, or else they would have done it in that sentence. So here's the sperm cell part. So here we got one of those. Here's a big picture. Now we're going to take a little part of it and kind of enlarge it. So here we have, again, another place in your body that's full of tubes. There's blood vessels in there also. There have to be that to deliver nutrients and oxygen and things to all these cells. But this set of tubes that a testis is full of are called seminiferous tubules. There's that word. So that's why I said they might ask you to diagram that. They're really tiny, and there's a whole bunch of them. So they took just one of them and cut a cross section of it to see that it's got a really thick wall, right? And then the tube part means there's a lumen. There's that word again, right? Lumen. So here now they took just one section of that thick wall along with the lumen there to show you how sperm cells form. So here, if we connect this diagram with this diagram over here, we see kind of like, and with the same language, at least the same root words, we started with an oogonium up here that was diploid and can do mitosis to make a lot more of themselves, right? With the right signals, some of those start to do meiosis. And when they get that signal to do that, we give them a different name, which was, again, primary oocyte in the egg cell formation. Now it's primary spermatocyte. If now we go back to this and we follow these two lines, we see that in this diagram, and they might ask you this, in this diagram, on the outer edge of these seminiferous tubules, of which there are yards and yards of inside a testes, lots of, in the very outer edge, you find those spermatogoniums. When they get the message to do meiosis, they also start to get pushed around and migrate toward the opening, where eventually what they develop into, sperm cells, are going to be released in that opening. So the egg cells, if you remember, in an ovary, they also take a kind of a physical journey around the ovary to a point in time and a place where they're right up against the edge of where they're going to be released, that single egg cell. Well, here it's a bunch of sperm cells, but it's the same kind of idea. As they go on this journey through the ovary or through the testes, they're also going through the stages of meiosis. So by the time it gets about halfway down, it's ready to start meiosis. By the time it's finished meiosis 1, it's a little bit closer. We call it a secondary spermatocyte. It's haploid. They might want you to know that, just like secondary oocyte is haploid, right? But with double-stranded chromosomes, awaiting meiosis 2, which here in males, that's going to happen right away. Like I said, in females, that doesn't even happen unless that secondary oocyte gets fertilized. But in males, it's just going to continue. They're also going to continue to move. Then this thing that we're going to call a sperm cell someday, when it grows up, we call a spermatid at first, and it's going to start to differentiate, which means it's going to grow a flagella. It's also going to do away with a lot of the rest of its organelles, which it didn't have many to begin with anyway. These are very short-lived cells. They are designed by evolution to do one thing: deliver this haploid nucleus in a relatively short period of time to an egg cell or die. So once they finish all that differentiation in their actual sperm cells with a flagella that can move themselves around, that's when they have reached that part of the lumen, which means they're on their way. This is very kidney-like. Remember, about 1 million nephrons funnel into some tubes, collecting ducts and other tubes, and finally into one tube that's leading out of the kidney. This one tube that's leading out of here is called the vas deferens, the sperm duct. And again, vas deferens was the name in the syllabus. So there is the sperm cell, and that's another thing they might ask you to annotate, just like they did the egg cell. So here's the sperm cell, pretty simple here. You have the haploid nucleus. I'll put the haploid part in there. You have a centriole. They wouldn't ask you that. You have a mitochondria. They might ask you that. You have a flagella. They quite likely will ask you that. And that's a really bad for a biology book to label that as a tail instead of a flagella. But then you have something that they're also going to ask you, and that's acrosome. And we'll get back to what that does later, but it is pretty much like a lysosome. It is a vesicle, so that means it was formed by Golgi, which aren't around much in here anymore, if at all. In the early spermatogonium, the Golgi were forming this vesicle. It's got digestive enzymes in it, and like a lysosome, it's going to let them go, but it's going to let them go when this sperm cell bumps into an egg cell. I'm getting a little bit ahead of myself, but while it's here, that is a name that you would need to put on a label diagram, or be expected to identify that this is, it's not a vacuole, it's not endoplasmic reticulum, it's not any of those cell parts, and it's not the nucleus. It's right at the tip, and it's the acrosome. Okay, so now we kind of switch gears a little bit and get to probably the most complicated part of this is the menstrual cycle, because they have very specific chemicals involved. They're going to have more specific ones when we get to the purple print later on. We have the idea of negative feedback, but also positive feedback. And this is where this gets pretty tricky, more than most other systems we've had that involve some kind of hormone and negative feedback, things like that. We have hormones that are made by the ovaries, and then we have hormones that are made by the pituitary, and you can see they want you to be able to differentiate them and know their exact names. So here they are. These are the pituitary hormones, and these are the ovarian hormones. And the role in the menstrual cycle of all these is expected. So we're going to break that down, and this is like I said, the trickiest part of this. So here, what they didn't add, and I hesitate to say that because I'm going to wait to see more purple print, but there's another hormone that comes from the hypothalamus that sends a message to the pituitary to release these, which sends a message to the ovaries to release these. We have seen this before. One kind of hormone triggers the release of a different kind of hormone from a different gland, which triggers the release of a different kind of hormone from yet another different gland. And so this is not an unusual kind of thing. We've seen it before. Okay. This just, I put these words here. You can read. I'm going to wait till we have a diagram up here in a second, because I've already identified that these are the ones that are secreted by the ovaries, ovarian, right? These are the ones secreted by the pituitary, and they just have their initials in the syllabus, so they won't ask you these names, but that's what they stand for. And then this is the one I told you is coming from the hypothalamus, which really kind of starts the show, so to speak, if you want to call it that, which they didn't put in there. So here's a very common kind of graph. In fact, it's 4 graphs in one, as you can see, and they're stacked on top of each other because they're all showing different events that are happening concurrently and have effects on each other. Here's the hormones that are coming from the pituitary and how they fluctuate across this cycle, which is always represented as 28 days, even though there's some variation there. The conventional way to show this cycle is 28-day cycle. Then they show the ones that are coming from the ovary and how they go up and down in concentration, fluctuate, right, in relation to these hormones up here from the pituitary. Then they show you what's going on physically, even with pictures, in the ovary itself, specifically in the follicles of the ovary, which were also in that other diagram, right, of the ovary when these follicles were getting bigger and bigger and finally released the egg and then became the corpus luteum. So you've seen those pictures before. And then this is that lining of the uterus, which is so involved, called the endometrium. And so all of these events are coordinated in conjunction. I didn't know exactly where to stick this in, but there it is. This is in vitro fertilization. That means a doctor took the egg cells out of an ovary and put them in a petri dish with the right soup around it, so to speak, and put the sperm cells on top of it in there. That's what in vitro means. In like a test tube in a laboratory, the more natural internal fertilization called in vivo is inside of a body. In vivo is inside a body. The use of drugs to suspend the secretion of these hormones, followed by the use of artificial doses of them to make a whole lot of eggs, because in a human, which is really almost only where you do this, although you might do it for livestock and things like that, but to go into an ovary and get these eggs, you want to get a bunch of them, because all these processes done in a laboratory don't have an incredibly high success rate. So you don't want to just go get one egg cell and all the surgery that it takes to do that. So you artificially induce with high doses of these hormones, the ones that cause actual ovulation, which we're getting to in a minute. And then you want to get those eggs to put sperm cells on IVF style in a laboratory and establish pregnancy. So while they're up there, because once again, I forget what's on the next few slides, let's look at this. If we start at day 0, now what does day 0 mean? Well, this cycle is determined by day 0 means that's when menstrual flow has started. So if we go down here, we see that this is where a uterine lining that has grown quite thick and has lots of blood vessels in it has been shed. Boom, that's what is called menstruation, right? And so if we start the cycle right at the shedding of that, and this is conventional, right? We see that in the ovary, there's a lot of primary follicles. They're going to trace just one of them, but one of them now gets the message, okay, it's time for you to start growing. What's that mean? All kinds of cells are doing mitosis, right? It's time for some of those cells to start doing meiosis. Just a small handful start doing meiosis. And as this follicle grows and grows, what its goal is, almost, if you want to call it that, is to have that secondary oocyte, remember, actually released. That moment called ovulation is obviously a very key moment here, because locked up inside this ovary, inside this follicle, no sperm cells are ever going to reach it. It has to be released. And so this is a key point. And if it is a 28-day cycle, that's going to be on day 14 when ovulation happens. Now we go to these hormones, okay? From the pituitary, right after menstruation, we go back here, the level of these two hormones is kind of steady, LH and FSH. It's kind of low, it's kind of steady, but what they're telling the ovary to do is to make more of these. So gradually those cells get the message. What does this mean is happening? Gene regulation. They're getting the message that says start to use those genes that make estrogen and progesterone. Now, back in review, estrogen and progesterone, as well as testosterone, are lipid hormones. So the genes aren't directly instructions to make them, because genes are instructions to make proteins, but the genes that are getting turned on are the genes that make proteins and enzymes that will form estrogen and progesterone out of what? Remember? Cholesterol. So that kind of story factors in here too. So in order to make these hormones, you need cholesterol, and for a male to make testosterone, you need cholesterol. So anyway, you can see that the estrogens really start to go up in the early days, you know, for like after menstruation, a week later, 2 weeks later. And estrogen basically is a growth hormone. It's causing the growth of, actually, I'm going to take that back. It's causing the growth of this endometrium, the estrogens are, but what's causing the growth of this follicle is the aptly named follicle stimulating hormone, the red one. This is causing the follicle to grow, but it's also causing it to produce a lot of estrogen. So the estrogen level goes up, up, up. And now you can see something that is a tricky part here, which we're going to see in a little video, but I'm going to show it here. Okay. Estrogen level goes up, up, up. This sounds like negative feedback is on its way. When things go up, 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 the increase itself starts the trend to make it go down, down, down. And that's why the levels of these stay kind of low, because the increasing estrogen does have negative feedback effects all the way back on the hypothalamus, but we're going to talk about just the pituitary because they left the hypothalamus out, right? Until it reaches really high. And this is the tricky part. When it reaches really high, it turns around and triggers just the opposite. It turns around a positive feedback response, which causes it to go really high, and the really high amounts of estrogen make this happen. A spike in LH, specifically LH now, we're focusing on that. But in both FSH and LH, their levels were kept low by negative feedback for this first part of the cycle. The increase in estrogen caused them to stay where they were. But then when estrogen gets really high, it triggers these pituitary cells to make a whole lot of LH. And this spike in LH is what triggers ovulation. This is the key event metabolically that triggers that very key event reproductively. Okay. So now, Now, what does this really high spike do? Now we get the negative feedback again. It causes the pituitary to say, okay, I made enough of that. I made it really fast. I made enough. Let me make a whole lot less. So FSH also, they go back to kind of a baseline level. Okay. Now we get our hormone that we haven't talked about yet, because up until now, it really hasn't been around hardly at all. But now, again, the ovary is what makes it. You got to keep that straight. Now, this ovary, specifically this burst follicle, now called the corpus luteum, it starts to secrete a lot of this. And our estrogen levels are also pretty high, because what this tells the ovary and this to keep making is more estrogen. So now this burst follicle, the corpus luteum, makes a whole bunch of these, because now here's the message, okay? We might have a baby on the way. We just let go of a secondary oocyte, right? And if it gets fertilized, it needs to implant to get nutrition from mom in the wall of this ovary, so that mom and this growing embryo, soon to be fetus, can get nutrients and exchange waste. So the last thing we want to happen is what happens when a big drop in estrogen and progesterone happen and this lining gets shed. We don't want this lining shed if there's a little baby to be attached. So they stay high for a while. This corpus luteum keeps producing progesterone and estrogen for a while. But if a little baby to be, which we're going to get a name in a second, I'll call it a blastocyst right now. That's what you were named before you got a name. If this little blastocyst doesn't implant and start secreting its own message, like, hey, I'm here, which is coming up in a minute in the syllabus, then this corpus luteum, it's almost like it wears out. It's in overdrive, making these hormones, and these cells, in a sense, kind of wear out in a simple sense, and they start doing that apoptosis thing. They stop producing so much progesterone and estrogen, and that is the signal for all of this to get shed and all of these cells to do apoptosis, and now we're back at a new cycle started. So that took a while and that was messy, but to have these graphs here really helps to show the interplay of those hormones. So now we'll get it in some syllabus words as well. Well, I just threw this in here. It could be anywhere, but this is another male-female difference. And now there's things that could go wrong, but males theoretically can keep making sperm cells their entire life. But females, all this activity and all these organs and cells and things, they again kind of wear out. Now, evolutionarily, this makes sense, because as a female gets older, her chance of successful childbirths go really, really diminish. And so all this energy now put into that, with the possible death of not just the embryo fetus, but also mom, that would be something you would judge to be non-adaptive. And so what evolution has resulted in is say, okay, ladies, when you get somewhere around here, it's time to shut down your reproductive systems, and your ovaries are no longer going to listen to this talk that's coming to them in the form of these hormones that tell it to keep making these other hormones, estrogen and progesterone, and keep producing these eggs. So that's what menopause basically is in a nutshell. Menopause didn't make the EOC list. Yeah, I had to throw that in there. Sorry. This is really neat, but it's Photoshop. Sorry to disappoint you, because it looks like it's really neat, and it's, theoretically possible, I guess, but I've come to understand it's Photoshop. So I'll give you a second to digest that one, because that's good from all different kinds of angles. That's pretty good.